It's a real honor to be here at the Women's Brain Initiative. And I'm going to talk about something completely different, brain circuits, mediating the generation and hot flushes. And to do that, I'll start with a coronal section of the human brain. And so you may recognize some of these landmarks: the lateral ventricles, anterior commissure, optic tracts. The hypothalamus is at the base of the brain surrounding the third ventricle here. And I've drawn these two arc-shaped nuclei. These are called the infundibular nucleus in the human, arcuate nucleus in other species. And in the 80s, Ernest Nobile and his colleagues dropped multi-unit electrodes down in this area and recorded pulses or volleys of electrical activity that were precisely timed with pulses of LH in peripheral plasma. And they named this pulsatile activity generator the GnRH pulse generator, and with other studies defined this area as the control center for reproduction in the primate. And they did this in the rhesus monkey. Now, a decade later, when I became a neuropathologist, I took advantage of my access to human brains, and I wondered, what would happen to human brain when women go through menopause? Could we see any difference between the hypothalamus and pre- and postmenopausal women? And this is the area I looked at. And indeed, there were some really amazing changes. Observing these changes was really a career-defining moment for me. Here we see a Nissl stain of neurons before the menopause in the infundibular nucleus and after the menopause. And you can see there are some very big neurons after the menopause in postmenopausal women. So there was this hypertrophy of neurons, and we were just amazed to see this. Even more important, the hypertrophied neurons we showed express estrogen receptor messenger RNA. So these weren't just any neurons that were getting larger in postmenopausal women. They were securely tied to the reproductive axis. They were target tissues for estrogen action. And because of the presence of estrogen receptor messenger RNA, because they were getting bigger in postmenopausal women, we hypothesized that the neurons were getting larger because of ovarian failure, of menopause, and estrogen withdrawal. Now, something that's very important to know about these hypertrophied neurons is they express the mRNAs for neurokinin B and kisspeptin, neurokinin B and kisspeptin. So here is in situ hybridization for kisspeptin in this case, and you can see this small wimpy little neuron in the premenopausal women. The grains mark the location of messenger RNA, and these neurons in postmenopausal women that were twice or 3 times the size, they had more grains, they had more messenger RNA expression, really a remarkable increase in gene expression. Now, this is very important, and perhaps more than anything underscores the importance of these neurons to human reproduction, because both neurokinin B and kisspeptin are essential for human reproduction. If you have humans that have mutations in either the neurokinin B or the kisspeptin gene or their receptors, they develop hypogonadotropic hypogonadism. They do not go through puberty. This is both male and female. They do not develop secondary sex characteristics, and they are infertile. So this, more than anything else, ties them securely within the reproductive axis. It's also important to note that ovariectomy of young monkeys could produce identical findings. Again, strong information supporting our hypothesis that the neurons were getting bigger because of ovarian failure. So let's place these in context. So here we have a neuroendocrine circuitry in postmenopausal women. We have ovarian failure, which leads to loss of ovarian estrogen secretion. And because of lack of estrogen negative feedback, we have a corresponding increase in LH and FSH in peripheral plasma. Now, I've also drawn these really large neurons in this circuit. They're called candy neurons because they have kisspeptin, neurokinin B, and dynorphin. So the kiss is where the K comes from, the neurokinin B is the N, the DY is dynorphin, so they're called candy neurons. And the gene expression is more remarkably elevated, and they have estrogen receptor alpha. They also project to GnRH neurons in the medial basal hypothalamus. GnRH messenger RNA is also increased in postmenopausal women, but this isn't a separate population of neurons in the human hypothalamus. And this goes along with the idea that the reason LH and FSH is up in postmenopausal women is because of increased GnRH secretion into the portal system that affects LH secretion. So one advantage of studying these neurons is they're remarkably conserved among animal species. They've now been described in the rat and the mare and the goat and the pig and the sheep, and there's been some absolutely incredible studies in the sheep on these neurons. And we can really take advantage of the animal models to be able to discern their function. And I think there are many labs in Boston that have contributed significantly to this. I'm looking at Dr. Kaiser here. Contributed significantly into our understanding, and indeed that's true around the world. There's been a concerted effort to study these candy neurons. And I think it's safe to say that there is now a consensus emerging that these neurons are responsible for the pulses of GnRH that cause pulses of LH in peripheral plasma, that these neurons are most likely what Ernest Nobile was recording in the 1980s that was giving rise to that activity, that these neurons could in fact be the GnRH pulse generator. So now one of the things that we've been doing to study the function of these neurons is trying to get an idea of their neuroanatomy. And so we've turned to studies in the rodent to do this. So our studies in the rodent give you an idea of how these neurons could coordinate to give rise to pulses of LH secretion. So here's a schematic diagram from some of our tract tracing studies and immunocytochemical studies in the rat. This is a coronal section, third ventricle is here, the arcuate nucleus is on either side, and I've drawn in a GnRH neuron here, which is projecting to the median eminence where the portal system is. GnRH dumps its contents into the median eminence, which goes to the anterior pituitary gland and causes the pulses of LH. And I've drawn a candy neuron. If you focus your attention on this neuron in red, candy neurons branch extensively within the arcuate nucleus. They give rise to processes that cross under the median eminence, across the median eminence to the other side. They form an arbor that projects to other candy neurons within the arcuate nucleus on the same side, and they also send projections to, GnRH terminals in the median eminence. And this would explain how these neurons could be coordinated to send out a signal to give a precisely timed pulse of GnRH in peripheral plasma, that there is a mechanism for coordination between these neurons that can coordinate pulses of GnRH. The other thing I want you to notice is that these neurons have the neurokinin 3 receptor shown here in this diagram, the little symbol here. They have the neurokinin B receptor, which is the NK3 receptor. And so they can communicate with each other via NKB signaling to coordinate pulses of GnRH. Okay, so that's all well and good, but what has this got to do with hot flushes? I mean, isn't she supposed to be talking about hot flushes? I mean, here you have a syndrome that affects millions and millions and millions of women, not only menopausal women, but young women taking tamoxifen for breast cancer, men getting anti-androgen therapy for prostate cancer, young women with premature ovarian failure. And when I had my first hot flush, I wondered, why hasn't anybody figured this out? Because flushes, if you read the textbook, flushes were an enigma. They said it was an enigma, or they'd say it's poorly understood, or it's a black box, or it's a pink box. Why didn't anybody figure this out? So you got to ask yourself, how are you going to figure this out? So I think the first question is, well, what exactly is a flush? So We know that flushes are caused by estrogen withdrawal. They're treated most effectively by estrogen replacement. And I think you can best describe a flush as an episodic activation of heat dissipation effectors. Okay, now this is where I lose the audience altogether when I start talking about heat dissipation effectors. But you know what heat dissipation effectors are. They're physiological mechanisms to remove heat from the body. That's why they're heat dissipation and they're effective. Because if you really trigger your heat dissipation effectors, you're going to lose your body heat. And that's important. Body temperature is very carefully regulated. You don't want to go too many degrees up or the enzymatic activity goes a little haywire. So you know what these are. They are vasodilation, which literally is the cardinal sign of a menopausal flush. When you vasodilate, the blood comes to the surface, and you can measure the hot blood coming to the surface by just putting a thermosensor on the surface of the skin. Okay, and that's a way to remove heat from the body. Sweating and behavioral effects. Behavioral effects, probably the most effective heat dissipation effector in human. So here is these are studies by Robert Friedman showing skin temperature during a menopausal flush. And you can see this rapid rise in skin temperature due to the blood coming to the surface of your skin. And that is so effective, it's causing a drop in core temperature. So we can think of flush as an activation of heat dissipation effectors. Now, a clue that these could possibly be due to the candy neurons comes from classic studies by Casper and Yin, where they recorded pulses of LH secretion along with hot flushes. So each of these arrows is the side of the flush. This is in 3 women, and they are measuring plasma LH and peripheral plasma every 2 to 3 minutes. And you can see there is the flush here, and just immediately after the flush is the peak in LH. But the LH begins to rise at the time of flushes, and the flushes are very exquisitely coordinated with pulses of LH. So I've told you that candy neurons are involved in pulses of LH. So why couldn't the candy neurons be involved with this? Now, it's important to note it's not GnRH that's causing the flushes, because you can take somebody with Kallman syndrome that don't have any GnRH neurons, you replace them with estrogen, you withdraw the estrogen, they'll still flush. It's not LH that's causing the flushes, because you can take a woman who has lost their pituitary, say from Sheehan syndrome, and you replace the estrogen, withdraw it, and they'll still flushes. So why couldn't it be someplace up in the brain that could cause the hot flushes? Well, it's a very intriguing hypothesis, but there was really no evidence in any species that candy neurons had anything to do with thermoregulation. So that's what we set out to do, to see if there was some connection with candy neurons or NKB signaling with thermoregulation. And the way we did this is we started with a rodent model. So we've studied both rats and mice. Here's a mouse here. And these are data loggers that are mounted on the surface of the tail. So we could put these data loggers in this plastic housing, put it on the tail with a little adhesive tape or whatnot, and these mice would just, behave normally, and we could monitor tail skin temperature, and we could also monitor core temperature through telemetry. Why did we monitor tail skin temperature? Because the rest of the mouse is covered by fur, that the tail operates as a heat exchange organ, and so tail skin temperature rises when heat dissipation effectors are triggered. What we found is that estrogen had a very powerful effect on tail skin temperature, this time in a rat. So this ovaryectomized tail skin temperature here is at the top. And when we replace estrogen or estrogen progesterone, we got a decrease in tail skin temperature in parallel with estrogen's effects on flushes in humans. So we were ready to do our experiments. But, at this point, it was like, okay, we know it's heat dissipation effectors. Where do we look in the hypothalamus? But fortunately for us, there were classic studies performed by Nakamura and Morrison in 2010 that described the circuitry for heat dissipation effectors in the rodent. So in a nutshell, it starts with the skin, with environmental warming. Then there is a multisynaptic pathway that goes up to the hypothalamus. And the first place in the hypothalamus that it reaches is the median preoptic nucleus, the median preoptic nucleus. Then there is a multisynaptic circuitry that ends up, you go through skin vasodilation, and if it works, the temperature will drop. Here I have drawn this a little bit more simply. Again, it's warm signals from the skin relaying through the preoptic hypothalamus to an effector arm regulating heat dissipation effectors, and you'll get tail skin vasodilation and decreased core temperature. So now I have superimposed on this drawing the candy neurons. I've drawn the base of the brain. Here is the optic chiasm. This is a sagittal diagram. The nose of the rat is here, the tail is there, and the pituitary is here, and the candy neuron is just above the pituitary. So I've drawn that in red. And the candy neurons we found from our track tracing studies projected to the median preoptic nucleus, which is an important part of the heat dissipation pathway. Not only that, medial preoptic neurons express the primary receptor for NKB, the neurokinin 3 receptor. So this was very exciting because at last we had a way to conceptually think about how candy neurons could influence the heat dissipation pathway. So, once you have a framework like this, you have an anatomic pathway that explains a lot, but it gives you a framework for hypothesis testing so that you could inhibit or stimulate different parts of the circuit and see if it was actually a functional circuit. So, for example, one of the first things we did, and shown by this lightning bolt here, is we put a cannula right down into the median preoptic nucleus and infused a neurokinin 3 agonist to see if we could activate this circuit. And indeed, we did get a very profound decrease in core temperature shown here. So you can see this is the control. There was a little rise in core temperature from handling the rat, and the ones that had Senkide, the NK3 agonist infused in, had a very profound loss in core temperature, indicating we had activated the heat defense pathway. We have also destroyed the neurons here and then found that even if we give a subcutaneous injection of Senkide, it has no effect. So these neurons are very important for Senkide, this NK3 agonist, produce this triggering of the heat defense pathway. More recently, we've done some studies in the mouse where we've injected the agonist subcutaneously, and you can see this rise in tail skin temperature and the drop in T-core, and doesn't that look like the studies of Robert Friedman where there was a menopausal flush in women? In Robert Steiner's lab, Ashley Krull has recently published a study where they injected Senkide subcutaneously, and they put the mice in a thermogradient. So a thermogradient, here you can see the thermogradient, the temperature was 25 to 45, and the mouse could choose anywhere on this gradient to run. And what the mice did with Senkide, the vehicle, they ran a little bit towards the cold, but with the Senkide, the NK3 agonists, they really ran towards the cold. And that's the closest thing you're going to get for a mouse telling you that they're hot, okay? That mouse said to us, it's hot, I'm running to a cold environment. So we have three different symptoms of the menopausal flush induced by this NK3 receptor agonist. Well, what has this got to do with the candy neurons? Well, of course, we had to do something to show the candy neurons involved. So what we did is we got rid of them, we ablated them. And the way we ablated them is we used a compound called NK3 sap, What this was an NK3 receptor agonist that was conjugated to saccharin, which is a neurotoxin, and it will bind specifically to neurons that have the NK3 receptor. Remember, I told you had to remember that these have the NK3 receptor. So the NK3 sap is internalized and it destroys these neurons. So here we see the control side and the NK3 sap. These are the candy neurons, this immunohistochemistry for NKB. You can see all the NKB neurons in the control side, and they're lost in the NK3 sap injected side. These were, was injected right into the, well, these control side. We injected it bilaterally in the ablated animals. And the specificity of this effect was shown by neuropeptide Y immunohistochemistry, shown here, that there was no difference between the control animals and the NK3 saps. And that was also true for POMC and GnRH terminals. So what happened when we did this? First, concentrate on the control side. You see I have the tail skin temperature of a rat. I've drawn it in hot pink because they're flushing. No, they're not really, but they're hot. And then it's reduced by estrogen. And then when we ablated these neurons, there was a very profound decrease in tail skin temperature, and it was no longer regulated by estrogen. So this is evidence that candy neurons facilitate cutaneous vasodilation, and this is a cardinal sign of a hot flush. So this is very good supporting evidence that candy neurons could contribute to the generation of flushes. Now here I've put the two systems together, the control of LH secretion and the interface with the heat defense pathway. So you've already seen the heat defense pathway here in this diagram. We have the candy neurons again in red, and they do branch within the arcuate, project in the medial preoptic area, median preoptic nucleus, and a separate branch connects to the GnRH terminals in the median eminence. And I think this really shows how candy neurons, which are responsive to estrogen withdrawal, they have estrogen receptors, could be the interface that would integrate the reproductive axis with thermoregulatory system. And it makes perfect sense that these would be integrated. After all, in pregnancy, you would not want the temperatures of the mother to go too high because you would be risking compromise of the fetus. So it makes perfect sense that reproduction is integrated with thermoregulation, just like reproduction is integrated with other systems such as metabolism and nutrition and stress. It also shows, if this is correct, it also can explain why Casper and Yen were able to show that pulses of LH were timed with pulses with hot flushes if this neuron is responsible and projects to both areas. Now, a very interesting paper just came out in 2008 by Stephanie Padilla in Richard Palmer's lab. And what they did is they used a transgenic mouse that had a kisspeptin-Cree mouse, so they could induce channel rhodopsin into the arcuate nucleus. See, there's the arcuate nucleus in the preoptic area that we've talked to before. Excuse me. This is their graphical abstract taken directly from their paper. And so they could do, by inducing this channel rhodopsin into the candy neurons, they could optogenetically stimulate them. And they found when they stimulate directly in the arcuate or even in the terminals, the candy neuron, the terminals in the preoptic area, they were able to induce a hot flush, which is rise in tail skin temperature, here shown by thermal imaging. If they were to infuse a neurokinin antagonist into the preoptic area before the stimulation, that effect was lost. So I think this is a really good evidence that we do have a functional circuit, and now it's been confirmed by another laboratory using very different methods, and I have to say this is a very elegant study. But you can do studies. Oh, I was going to say we recently are more trying to refine the circuit, and we recently have found that these neurons in the median preoptic area have glutamate. So they're excitatory neurons, and we're starting to refine the circuitry even more. We have found that these candy neurons don't project directly to the heat dissipation pathway. We now have evidence that it's an indirect effect via these NK3 neurons that have glutamate, and those project to the heat dissipation pathway. So the diagram that I've shown you, we're going to have to be modifying that, and I think a lot of people now are starting to study this, and we'll see other modifications as well. But you can say, well, this is all very nice and good. You can make a mouse have a hot flash. What does this got to do with humans? And I can now say that there is some very compelling evidence that this hypothesis may be correct, that candy neurons could generate hot flushes through NK3 signaling through the preoptic hypothalamus. And one of the a very important study was done by Carolyn Crandall at UCLA, published in 2016, and she did a GWAS study with 17,695 subjects and found that there were 14 SNPs associated with menopausal flushes, and they were all in the TACR3 gene, which is the gene encoding neurokinin 3. So that's pretty convincing that evidence that perhaps we're on the right track, and maybe information like this would explain why some people get menopausal flushes and some people never do. Now, another study was done by Wajid Dillo and his colleagues in Imperial College London, and this is the title of their paper, and this is my exclamation point. Neurokinin B administration induces hot flushes in women. They had 10 women, and they infused either vehicle or NKB, and you can see these little red dots are where the women reported hot flushes. So, and these were backed up by physiological measurements. And finally, there is now a lot of clinical interest in treating hot flushes with neurokinin B receptor antagonists. This is the results from 2 such studies. We have Julia Prague and Wajid Dillo's study on the left, and Graham Fraser gave me this on the right. This is called MLE 4901. This is called fezolinetant. This shows the change in hot flush frequency from baseline. You can see how effective these compounds are. This compound is equally effective with this other one. What's remarkable is that the NK3 receptor agonists reduced hot flushes within 3 days, and they were kept on for the whole study here, 12-week, with 40 different individuals, a very impressive reduction of hot flushes, certainly as effective as estrogen, and also acting much faster. So this is the status of all the studies that are going on now. There are 4 companies involved. Melendo, who Prague and Dillos published the first information on, has discontinued this because there has been an elevation in liver function enzymes. There are now 3 other compounds in the works. One of these is an NK1 and NK3 agonist. The other ones are NK3 agonists. The 3 out of the 4 that have been tried have been very effective against hot flushes. Phase 3 trial has now been initiated for this one. Phase 2B is now for this one. And this other agonist, they found that it reduced LH secretion in men, and undoubtedly that will probably be, is compelling information that it will also be effective for hot flushes. I think the thing that's so remarkable is they all very effective, but they're all different compounds. They're all tested by different individuals. And so there's a lot of hope that one of these will make it to the clinical arena. So the efficacy of these is better than the other non-estrogenic products, and they're given orally. So this is the last slide to bring you back to really what instigated these studies is finding these hypertrophied neurons in postmenopausal women. And so I want to remind you that this is really based on the human autopsy studies that we know in postmenopausal women that these women have larger neurons. And it was really just kind of finding out what the physiological function of these neurons that led me to the hypothesis that they have hot flushes. And so I need to alter my diagram and put in this hypothesis that the hot flush is caused by preoptic neurokinin 3 neurons that modulate central thermoregulatory pathways. And so far, the neurokinin 3 antagonists look promising, and perhaps they are going to be at the sweet spot because they are both on the KNDy neurons and in the preoptic thermoregulatory areas. But it's only by looking at the microcircuitry or the circuitry that could cause hot flushes that we can begin to develop rational treatments for hot flushes that do not involve estrogen replacement therapy. And so I really think that we should stop talking about flushes like they're enigma and really start to talk about what are the preoptic thermoregulatory pathways that are impacted by neurons that have estrogen receptor or impacted by estrogen withdrawal. And that's the way we're going to move this field forward to really understand the mechanism of hot flushes and then figure out how to adequately treat them. So it's very important for me to acknowledge the many people that contributed to this work. In particular, today I talked about the infusion studies of Penny Dax and Melinda Middleman-Smith, but all the graduate students have been so wonderful throughout my career in contributing to work. And I also want to acknowledge Sally Krajewski-Hall, who's been in the lab for 20 years and really is the goddess of immunohistochemistry in our lab. Nathaniel McMullen, who is my collaborator, is a neuroanatomist, and he is also my collaborator in life. He's my husband. And Andrew Romanofsky, who, and if any of you decide to go into looking into hot flushes, I highly recommend collaborating with somebody like Andrew Romanofsky, who's a basic thermobiologist, because he really did have to explain many aspects of the thermoregulatory system. And I think, getting reproductive biologists and thermoregulation experts is really the way to go. And finally, National Institutes on Aging and other basic funding for research. And I would say that it would not have happened without basic research from the National Institute of Aging. And really, this has been basic research from the postmenopausal human brains to all the rodent studies and now finally to the clinic. And you should be nice to your basic scientists. And, you don't know where it's going to take you. 10 years ago, maybe I would have said, well, I'm studying the temperature of a rat's tail, and they may not have appreciated how important it is. So keep that in mind when people describe their studies to you. So thank you very much.
Thank you.
Very elegant talk about the mechanism. I have two questions. One is when you treat with a synthetic antagonist versus an estrogen, are they both targeting the heat increase versus the reaction to that, which is the flush? Because you could think the flushes could be a good thing, right? And the real problem is that the heat goes up in the first place. So they both target the same thing.
Oh, I'm sorry. The question was...
So there are only two phases.
Well, with
one
the...
is the temperature goes up, and that's probably a bad thing.
Well, the core temperature goes down with the flush.
I know. So I would think the flushes could be an adaptive thing, a good heat dissipation You want
Yes.
to actually do that. So what you really need to do is prevent the heat from going up. Because
Well,
in the first place,
what's going up?
temperature, core temperature.
Oh, I'm not convinced that temperature goes up before a flush. I don't think, I've looked at Friedman's, Robert Friedman has some data showing that the temperature goes up immediately preceding the flush, if that's what you're talking about. I'm not so convinced
So you don't
from,
think the flush is a reaction to the heat going up, that you're trying
no,
to dissipate the heat?
well, the flush is, yeah, I don't think Robert Friedman had women swallow telemetry pills to determine what the core temperature was before a flush. And only in about 50 to 60 percent of the flushes was there any change in core temperature. And it was so small, it was not nearly what you'd expect to be a physiological trigger for the heat dissipation. So I'm not convinced that I don't think it's triggered because it's an inappropriate triggering of the heat dissipation effectors rather than the temperature going up before it. And there's been another study, I can't remember the particulars again, to say that it's not a response, the hot flush isn't response to increased core. You'd think it would be, but it's not. The skin is hot, but that's not reflecting the core, because the skin is hot, blood is rushing to the surface, and then the core drops. But that's not the core. The core goes down. So the
Yeah, I
core
know,
is,
this core goes down because you're cooling
yeah.
yourself because of the dilation.
But the trigger for the flush, there's a lot of different triggers for the flush, hot beverages, spicy foods, LH pulse, but it doesn't look, and maybe in some circumstances, but in general, I don't think it's triggered by increased
So then you don't
core.
think this, the second question was, you don't think this has anything to do with changes in core temperature during ovulation?
No, I don't see the connection there. I don't see the connection there. I think maybe, if anything, a heat dissipation of response would be very important in pregnancy because the fetus might be very sensitive. And there are antipyretic effects of pregnancy that the temperature, there may be a limitation on how high the temperature goes up. And that may be because this is the system that's trying to effectively have it go down. But I don't know, I would invite the audience to figure out what the physiological role of this activation of heat dissipation effectors are, because I think that's a really important question.
Also two, but hopefully quick questions. One, I might have missed this, but why do the neurons get bigger?
Oh, good.
And second, what is it about exposure to estrogen that is required, right? Because if you have no exposure to estrogen, you don't get hot flushes. So obviously it's having some sort of cellular effect that the withdrawal then
Yeah.
results in the flushes. So why is that?
Why are the neurons getting bigger? That's a good question. I always imagine in my mind it's because they're more active, because the neurons are getting bigger, the pituitary gonadotrophs are getting bigger. I mean, you've got increased LH and FSH, that it's more metabolic demand, but have I studied that? I have not. And I think there would be some very interesting questions at a molecular level that you could ask in terms of basic structural changes. What was the second question?
You need to have exposure to estrogen.
Yeah.
So how is that changing, presumably, the connecting neurons in such a way that then the subsequent withdrawal results in the...
No, that's a very good question. I think a related question is, why do women adapt over a period of time? So those type of questions, I think, are really things that somebody needs to look into.
Naomi, thank you very much for the very nice presentation. So as you know, most candy neurons are glutamatergic. So do you think it is possible that candy neurons are acting directly on these neurons in the perioptic area through glutamate rather than NKB in that population of neurons in the perioptic area? Could it be that NKB is only acting by reactivating candy neurons and then through glutamate activate those neurons?
Yeah, that's a good question. If you look at Stephanie Padilla's paper, when she stimulated the candy neurons, it was blocked by a neurokinin antagonist. So that would imply that the NKB was the important part there. And again, our Senkai does activate it quite readily. As well as far as whether glutamate could do it as well, I don't know. I haven't studied that, but we have found at least that NKB is the important component. And also, I think that's backed up by the studies in human now with the GWAS study showing that there's a lot of evidence that NKB.
Yeah, no, that I'm definitely not
But
opposed to
other
that.
things,
Like, where is NQB acting? Could it be acting mostly at the level of candy neurons, and then with
oh
these
yeah.
activations is to through glutamate activate the other area, rather than NKB acting in the preoptic area?
Oh, well, I think it's, the other question that's related that, again, I think it's, our evidence is that NKB is acting at the preoptic area, but that doesn't, I don't exclude anything else, I'm not, we studied the GnRH system long enough that knows that there's not just one thing controlling, right? So it certainly could be a factor, something I haven't addressed. But the other question is those NK3 receptor antagonists, are they working at the level of the candy neuron, or are they working at the preoptic area, or both? Or maybe that's the NKB is the best, NK3 is the best target because it could be two areas that they're acting on.
We have one more question.
Hello. Hi, my name is Heather Hirsch. I'm a menopause specialist. So my question would be, we know that when women go into menopause and they lose their endogenous estrogen, they start developing hot flashes most commonly, but they also start developing other common symptoms like sleep disorders or mood disorders. And so I don't know if there's any anecdotal data or if anyone's looking at using the antagonists to see if it improves not only hot flashes, but some of those other, parameters of sleep, mood, other major midlife changes.
Yes, Julia Prague in her study in Lancet did analyze sleep, and and I think the other compound as well improves sleep. And it's not clear if this is because it's an effect solely through reducing hot flushes, because if you reduce hot flushes, you're gonna improve sleep. So that's an open question whether there could be a separate influence on the sleep. Whether it's separate from hot flushes. But so far, the clinical trials that I cited, sleep is improved well because you've reduced hot flushes.
That's great, because we see so much when you use estrogen, sleep improves greatly. So
Yeah.
that circuit would also be really
Yeah.
interesting.
And another, the one that is an NK1, NK3 antagonist, they're not pitching, but they're using an NK1 antagonist because they think it has separate effects on sleep. But that'll be interesting to compare when you have two antagonists versus one.
Well, let's hold other questions for the coffee break and thank Naomi once again.